All right, yeah. Tell me about everything.
Sure. I've been studying for the MCAT for the last month and a half.
Hmm.
Um, and so the last month and a half was mostly content. Big mistake, but I feel like everyone makes that mistake.
Okay.
And so I took my first full length this past weekend.
That was
It was
the blue.
yes, and I ended up doing Jack Weston for cars, just like you said.
All right. What was your breakdown?
Right. So my breakdown was 125 chem phys, 122 cars, 125. Actually, let me recheck.
Mhm.
Biochem, I think it was 126,
Okay.
and then psych social was 126. It was a 499.
Hmm.
So
Got it.
I'm just gonna double check real quick. Give me a second. Um, yeah. So 125, 122, 126, 126.
Got it. Okay. When is your test scheduled?
Right. So this was one of the first things I wanted to ask you.
Mhm.
My test right now is scheduled for September 6th,
Got it.
which is in around 2.5 months.
Hmm.
Um, Now my goal score, so to speak, is around the 515 mark. I
Hmm.
mean, you tell everyone 523. I watched one
Yeah.
of your videos. So that's technically my goal, but like I
Yeah.
would be content with a 515, let's say.
Hmm.
Um, and so I don't know. I don't know if there's correlation between the, for example, at ACT and SAT and the MCAT, I got a 34 in my ACT. So I mean, at least in high school, I wasn't bad at standardized test taking.
Hmm.
It was like a 50-10 SAT conversion, but yeah, that's really it. Cars is my worst section, obviously, but I would like to with you. I would like to probably work for now at least on improving biochem and chem phys.
Hmm. Got it. Yeah. So, yeah, I definitely think that that's possible. The blueprint tests are significantly harder than the AMC tests. So it's good that you got this score on the blueprint test because it likely means that your AMC score would be much higher. And it's really good to do the blueprint tests for practice. So what I usually do is I kind of separate the uh like the entire like kind of schedule into like three phases.
Mhm.
I go backwards from your test date. So let me do that for you actually.
So this was actually the question I was going to ask. I went a little off track.
Mhm.
Um, do like I'm trying to view this in a very like I guess through a humble lens, so to speak. I am fine moving my test date to January. I just don't know if I can really like I don't know if it's possible to get a 515 by my September date, and I am open to moving it to January. What would you suggest in
Uh,
terms of like perspective?
yeah, I think we have enough time to give that a worthwhile shot. And if it comes to be that, because like what I'm going to do is um like phase one will be like the accuracy phase, and phase two will be like blueprint or Altius tests, and then phase three will be AMC tests. So basically, once we get to the AMC tests, we should see like a score jump compared to the blueprint stuff. So if we don't see that, then maybe you would push it back to like January. But I think that right now, September is definitely doable. But we would be able to see, like, we'd be able to make the decision in time for you if you need to uh push it back or not.
Okay,
Yeah.
understood.
I mean, it's like, yeah, like you're at least, because a lot of my students are like applying this year and or this cycle, and
Hmm.
they're kind of in a hard spot because they can't really push their tests back. So, but you can, but you also are taking it, you are taking it the latest that you can take it in a year. So that's what you have, like another thing that you have working for you. So yeah, I'm gonna start making this schedule for you. So –
Okay. 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 Okay.
yeah, let's see.
100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. 100%. I will still ask though, you make your students for cars start with the AAMC Q packs, or how does that work?
Oh yeah, the AMC stuff. Um,
Right.
yeah. So here I'm sharing my screen. You can see it, right?
Great. Yes, I can see it.
So okay, so like that's your MCAT date. So I'm gonna go like a week like before. I think it's preferable to have the last full length someone takes like a week before. So this would be phase three. And so that'll be the AMC full length. I mean, it's the free scored one. I call that full length five because it's the
Hmm.
latest one. There's something called a blind review, which I'll go into. Um, and that'll be for that week. And then here would be AMC full length four, AMC full length three. Like if we're gonna do like, a test a week. Um, oops, length one, and then there's the sample full length, and then before that, then phase two, which would be like a blueprint or Altius full length. So let's see. All right, let's see. Okay, well, since you've already pretty much started like the blueprint full length, third party full lengths, maybe we can just have you start with phase two. Let's see. You said that you took this past weekend, right? Was it like a
Yes,
Saturday, like
it was Sunday.
Sunday? Okay. And this was blueprint full length one, I guess, right?
Yes.
Okay. Um, all right, so let's say that, do you prefer to do that? Well,
I was actually planning on taking my second full length next Friday.
okay. Yeah. The reason
Yeah.
I did Friday is just because I went backwards from the week, from the day
Yeah.
us. But okay, if we do this, okay, let me get rid of this stuff.
There's a fair chance the following full lengths I'm going to have to take on a Saturday. I
Okay,
just want to say.
sure.
But I guess we could keep it Friday for now.
Mm-hmm. Yeah, so okay, let's see. Two, three, four, whoops, five. Okay, so yeah, we're probably going to be able to fit five, either Blueprint or Altius full length, and then all six of the AMC full lengths, and have it be one test a week, which is good because a lot of times for students, like they have less time and they have to do like two tests a week, which could be a lot, but
Hmm.
like this can work for us. So let's see. Okay, so there's also, well, I guess the phase one stuff that I talk about is um so you're using Anki, right? Are you?
Yes. So here's the issue. Anki a day just itself takes me like 3 hours to get
Okay.
through.
Yeah.
Um,
You don't have to do it that way. Like I know a lot of
mhm.
do it that way where it's like you have like 100 plus cards that it tells you that you need to review or whatever. I'm guessing like that's why.
Yep.
Yeah, so I think Anki is best to just do a little bit of every day. I don't count it in terms of cards, I count it in terms of time. So I have students do 45 minutes to an hour of Anki a day. So you don't have to do more than an hour, basically. Um, because I know that that's how it looks like it, and it will say something like, yeah, you have hundreds of cards due, and that can take forever. But yeah, I think Anki is best to just do a little bit of every day. So just 45 minutes to an hour, I would say to do Anki. What do you think?
Hmm. I mean, sure. Of course, I'll listen to you. I'm not gonna argue what you're saying. I do think it's like it's become a pretty good way of me for just like reviewing stuff, as opposed to like going to a textbook or something, which I know you also recommend, but it's just more time consuming and harder for me to focus like on a textbook. And so I had a mix of like Jack Sparrow and Miles Down, you know, those decks. But I also have a deck which is strictly just self-created deck that has mistakes from UWorld and full lengths, just like a sentence filling in, you know, a term I didn't know. Do you recommend I prioritize that
Oh yeah.
over?
So yeah, when you review your tests, that's a good way to review them. That's what I did as well, especially for Psych Soc, since Psych Soc is like, you know, full of like kind of content information that you may not be able to get from any one source. So when you review your tests for Psych Soc, a lot of times, like, you know, if you see, even if you got a question right, if you see a term like in the choices or something that you don't know, just add that to your list of or to your cards, your review cards. Yeah, I think that that's good. In terms of priority, I think like with the shared decks to prioritize, um, generally, I guess so like Anki is best to like for like memorizing stuff, right? So for the sections that have the most that are the most content dependent, those would be the ones to that Anki would be really helpful for. And that's typically psych soc, like behavioral sciences, and for biochem, like the metabolic pathways. But you know, everyone's different. Everyone has different stuff that they feel like they can brush up on. But yeah, I would still say like 45 minutes to an hour of Anki. Like, of course, there could be days where you don't do, because I guess we haven't talked about like any of the other stuff yet, but um so yeah, okay. Let's talk about some of the other stuff. So you said you're doing UWorld as well, right?
Yes.
Hmm.
But that I just started. Like before my full length on Sunday, it was straight up Anki for 3 hours, then watching some video, like videos on topics I don't understand.
Yeah.
I would do three cars passages a day and then like 5 year old questions. But now I'm trying to do a section every day, like a 59 question stimulated section.
Hmm. Got it. So let's see. What I guess what content areas do you like? Do you have like any maybe like a running list of like content areas that you feel iffy about?
More or less, yes. A lot of it is physics.
Okay.
There's also a lot of chemistry related things.
Yep.
Um, that was so broad. I just realized. My bad.
I don't know.
It's like a lot of acid based stuff I need to brush up on. In
Lots of what
physics,
stuff? Say that.
mm,
Lots of what stuff? Say that again.
like acid base
Oh,
titrations.
yep.
Yeah. Um, and then in terms of physics, there's a couple different things. I mean, optics I have issues with, but yeah, there's a couple things in physics. But yeah, I think Chem Phys is going to be the bulk of what I will be asking you to help me with.
Got it. Yeah. Okay, that works. Yeah, because we can like spend like some parts of our sessions like going over some physics concepts or some gen chem concepts or something that'll be faster than if you just watch like a video on it or like did Anki cards for it. So that would be like, you know, like how there's like in psych social like maintenance rehearsal and elaborate rehearsal. Like maintenance rehearsal, like when you're like trying to remember something, so like or learn something, like maintenance rehearsal is going to be like what Anki is, where you just pretty much memorize the thing. But elaborate rehearsal is when we do like a deep dive and we go into it and we really understand it on a fundamental like conceptual level. And that's always better than maintenance rehearsal, but obviously we're going to do both. But yeah, like for some content that you want to learn, you could use Anki for the maintenance rehearsal, and then when we go over it like with a deep dive, that'll be the elaborate rehearsal. So okay, that's good to know. And for cars, like you said you're doing like a couple of or like a passage a day or something like that.
It was around like 2 or 3 passages a day, but it's crazy. Honestly, with cars, I took the blueprint half length before I even started the MCAT
Huh?
prep. I got a 123. My score decreased by a point. So yeah, I mean, I just feel like with cars, it's like I'd rather use you for the sciences, but yeah, cars is also an issue, obviously.
Got it, got it. Okay, cool. So yeah, we can go over some physics concepts and stuff and passages and all of that stuff. How are like the calculation type questions for you?
Um, I think the thing I struggle with the most, and it's ironic because it's like one of the first things taught in Gen Chem, but just like moles, calculation, stuff like that,
Got it.
stoichiometry, I do have an issue with that sometimes. So
Got
like solubility constant, that's a great example.
it. Awesome. Got it. Okay. So yeah, let's do this. Let's have you do okay, so I'm gonna start by writing like okay, let's do like a phase one thing. So Anki, 45 minutes to an hour, and then oh, and there's an AMC section bank. Do you have that?
No, I was just waiting to buy because it's a 6 month thing, right? I think, or it might be a year. Regardless, I will buy it.
Okay. Because okay, let's see. Um Trying to think. Okay, so I think what we can do is this. I'll start you off with like a short version of the phase one. So that's going to be the for the so the Anki. I mean, the Anki thing I'm writing just for phase one, but you're really going to do Anki every day, like pretty much.
Hmm.
Like unless you take like rest days and stuff, but Anki is always going to be a thing. But we're gonna start you off on like the section bank for the sciences, like there's like three of them. There's like psycho, CP, BB, 100 questions, some of them passage based, some of them discrete. And then AMC cars, that like whatever you're doing, I think 2 to 3 passages is fine. And let's put this through like for like the next week, and then you'll take uh your next either blueprint or Altius full length. Um, oh yeah, and then let me go over a blind review. So basically, let's say that so what you're going to do is you're going to write down which questions you got wrong without like try not to look at the answer, but just don't write the answer down. So let's say for CP, you got 10 questions wrong. Um, Write down what those questions are and then do a second attempt of the test. Um, Without the time constraint and just answer the 10 that you got wrong. And the purpose of this is to separate the questions that you got wrong because you really didn't understand them from the questions that you got wrong because you were on a time crunch. Because, like, if you're running out of time and stuff and you get something wrong, it's almost like a waste to look at the solution right away because you didn't get really a fair chance at trying to figure it out. So that's what a blind review is basically. And let's say after the blind review, out of those 10 questions that you got wrong the first time, now you just have 5 that are wrong. So that means 5 of them like you actually understand, and you get to do them. So it's like, you're actually putting in the work and you're actually figuring it out on your own. But then after that, let's say for the 5 that you're still getting wrong, you just do a regular review, which is reading the solution essentially. Um, and then let's say like 2 of them still don't make sense. When we meet, we can go over the 2 rather than like all 10 to be like really efficient. But also if something, let's say you got like a question right, but you're just curious how I would go about it, um, that's also something that we can do when we meet. But
Hmm.
that's what a blind review is.
Got it. When you say AAMC section bank, I
Mhm.
just feel like that's like broad. I mean, like how much should I be doing?
Oh
You
yeah,
know?
yeah. So let's see. Okay, so whoops. So there's 300 questions in all, and if I divide that by seven, I get 42.8. So like maybe like 43 questions. That's not very whole number, but some
Yeah.
like roughly, whoops, like I'll just write like roughly like 40 questions. Um, and yeah, you'll see that it's like so it's like yeah, it's passage based stuff for the most part, but there's also discrete stuff. So let's see. And each section has 100 questions, and I think that's like probably going to be like maybe 10, 12 passages total. I can look into that though and update this to give you like a number of passages. Let's say, let's see. Okay, so let's say that there's 12 total, then maybe that could be like, or sorry, 12 total for each, which would be 36 total in all. So maybe something like, let's see. Are you doing anything else besides studying? Like, are you working or anything?
No, I would like to do. Like this is the thing. The next 2 months before I even go to school in August, it's like my full-time job, so to speak. So
Are you still you're still in school?
yeah, I mean, I'm going to be a senior, so I have a year left.
Got it. What are you, yeah, where are you?
I go to the University of Miami, but I'm from New York City,
We're in New York.
Brooklyn,
We're in Brooklyn.
like the Sheepshead Bay, like
Okay.
Sheepshead Bay, Brighton Beach. Yeah. Hmm?
I'm in Bushwick.
Oh wow.
Mhm.
Okay, that's cool.
Yeah. So okay, let's see. So maybe so you said like cars 2 to 3 passages a day, like you're kind of doing. For the section bank, I'm putting down 5 passages. I'm gonna like afterwards like take a look at the section bank to make sure that I get so to make sure that I know what actual number of passages are in each section of the section bank. But if my estimate is true for 12 passages in each, then 5 passages I think would be good for you to do to finish it. Like it's essentially going to be something that you finish by that time next week.
Yeah.
Like, well, I'm having it like I guess you can even technically start it today, but I wrote it down to be started on the 20 tomorrow. So
Hmm.
and a week from tomorrow would be your full length. So just basically you finish it by that point. So I think that'll be like 5 passages, but yeah, I'll double check. But yeah, that'll be that. And after that, so like then after this, you're going after you do your full length and your blind review and review, like do that like the next day, like not on the same day as your test. And then it's pretty much your test, like a test every week, right? So all this time in between the tests, we can spend doing like UWorld stuff. In addition to car stuff, like the cars stuff will still be AMC passages. Um, but for the um sciences, it'll be like so like basically like you'll take your blueprint test and you'll 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 kind of find like okay maybe like I'm having issues with like this topic or like let's say fluids or something and immunology or something. So then, when we meet, we can kind of go over those topics, and then you can do like UWorld targeted kind of content stuff by doing UWorld passages and questions on those topics that we find that you're weak in from the blueprint or from the test that you take. And then, you could do the UWorld questions on that, and then we can meet to go over those UWorld questions. And then you'll take your next full length, and then you'll do your blind review and review of your next full length, and then we'll meet, we'll go over some concepts and stuff and strategies, and then you'll do UWorld questions on that material, and then we'll meet to go over that, and then you'll do your next practice test. Like, Zach, yeah.
Yeah.
Okay. So like that means you're essentially going to have like three types of days. A day where you do a full length, a day where you review the full length, and a day where you do UWorld stuff in addition to like AMC like car stuff. And I mean, if we run out of AMC car stuff, we can do Jack Weston car stuff, but that's kind of basically how we can set everything up. So, and we're, yep. Yeah, what were we gonna say?
I've seen different people say different things, but a lot of people say to leave the AMC stuff for the last month. The thing is with cars especially, my issue is like if I'm getting, if I'm scoring solo on cars, it's likely me applying the wrong logic. My issue is like if I start doing the AAMC cars and continue applying this like the same like way of thinking, isn't it just going to be me wasting material, so to speak, or
So, yeah, so I know that you mentioned that you really wanted to focus on like CP stuff, but we should probably do some car stuff as well, so you kind of know
yeah.
how to approach it. So yeah, we can probably spend like some time on that at some point. Probably, we probably don't need to do it like every time, but like at least once for you to kind of have an idea of how to go about the passages, I would say to do like for that. So then you won't be kind of like wasting the passages that you have. Which will be a good
Right.
passages. Like it'll be the Cars Question Pack Volume 1, Volume 2, and Diagnostic Tool. Um, but yeah, I think that's what do you think about that?
Yeah, I think that's probably a smart way to go about it. I was thinking to meet, 2 hours every week, but like next week, for example, maybe we can do 2 hours like the usual, and then 1 hour cars
Mhm.
sometime
Yep.
if that works.
Yeah, we could totally do that.
Okay.
So yeah, so I'll just so basically this will be this. Oh, and then I guess to be more specific about the world targeted stuff, maybe, the thing that you've been doing with like 59 questions, um, you know, it should take you like 1.5 hours. That would be a good amount to aim for, because that's like how much you'd get on the test, and
Hmm.
do three cars passages. So basically something like that. Yeah,
I'll be right back 1 second.
sure.
All right. And also, um, in terms of, I also saw that like there's a thing where you ask students to time like how much they're studying and
Oh yeah,
input that somewhere. Is that an actual thing or no?
yeah. It's funny because I guess like a lot of these things are from like a long time ago for me, so I
Okay. Okay.
kind of forget. But
Yeah.
yeah, so like because a lot of those things also like I guess I haven't like continued a lot with students, but sometimes
Oh,
so yeah, that's still a thing. So basically, like that's like a way for you to kind of get like a good, accurate idea of how many hours you're putting in. So yeah, so the idea is that when you start studying for the day, you start a stopwatch, and if you do anything other than strictly speaking studying, like whatever, going to the bathroom, eating, going on a different website, going on your phone, whatever,
mm,
you pause the stopwatch, and if you resume studying for that day, you resume the stopwatch, and you can put in this calendar kind of schedule, um, the amount of time that you spent each day, in hours on this bottom cell here, and then I can just uh put something like write something in like um in this to keep count of like hours, this week,
hmm.
like total hours. So yeah, if you want to do that, you can definitely do that. And you would just put in this bottom cell for this, like how many hours you spent studying.
Okay. And so, starting tomorrow, you're saying those three things. Do you do you think that's gonna end up being around 6, 7 hours, or you think it's gonna be less time spent for
We
like?
6, 7 hours, like you're out. Are you asking?
Hmm?
So when you say that, are you asking about the section?
I'm asking, yeah, about like the schedule itself.
Hmm.
Like you expect me to do that in more or less in how long? Like Anki for 1 hour, but then the
Hmm.
section bank and the cars.
Yeah, so that should be, let's see. Okay, so all right, I open up the section bank. It's actually 8 passages each, not 12.
Wow.
That's 24. So how about 2 to 3 passages? Just like for cars, right?
Okay.
And in terms of how much time it should take, well, I mean, if you're able to do like, oh, well, I don't know if you said this, maybe I said this instead, but when you said that you were doing the UWorld like 59 questions, how long does that typically take?
I do it timed, so it's an
Okay.
hour 30 minutes. The issue is sometimes like I run out of time
Yeah,
and I end up not even like trying on a question.
I see. Yeah, yeah. Because that could be I like wasting the question. So
Yeah.
let's do this. Let's have you do it like timed, but when you well, I don't know if Blueprint even lets you do this, but let's say you do it timed and you run out of time, are you still able to, I guess, continue and continue to answer questions and stuff, or does it like it's like probably tutoring mode or something like that, but
In your world, yes, you can. Like I can time it, submit it, see the score, but then like copy and paste it and then like do a section. Like Blueprint, I don't know. With full lengths, I don't know how that's
okay.
gonna work.
I
But
you know I think actually then we don't have let's not worry about time constraints for the section bank, the cars here, and the UWorld stuff because you're going to be doing a practice test every week, which is going to be timed anyway, and that pretty much is how you fix timing. I mean, I never I always had the phase 1 thing be untimed because phase 2 and phase 3 involve a lot of timed practice tests, which will kind of autocorrect timing issues anyway. So let's actually like have you not do things in between with the time constraint. You could still put in, like or try to do it in that time constraint like you have for UWorld, but I would say that if you run out of time and your other, like, if you run out of time and you're not given a chance to answer some questions, then it's fine to go over and answer those questions, regardless of how much time it takes.
Hmm.
So, yeah. So I would say during the week when you're doing, like, so this week, I guess you're going to be doing the section bank stuff and the AMC car stuff. Yeah, you don't have to do that like timed. And if you do it timed, you don't have to stick to that time. If certain questions take longer, then allow them to take longer, because you want to get the most like kind of bang for your buck with accuracy. And that's pretty much what we'll be doing. So like in between, in between your practice tests, I would say, yeah, just to 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 either do it with a time constraint that isn't really a full constraint, because if you run out of time, you can still answer questions, or don't worry about the timing at all. So yeah,
Okay.
because yeah, that's also why the blind review exists, because like if you run out of time and you don't get a chance to even like try a question out, like yeah, it's like wasting the question. So yeah, so I would say that basically, like on the days where you're not doing a test, yeah, not to worry about timing.
Okay. And one second, what was I gonna ask?
Mhm.
Oh, I forgot what I was gonna ask. But there's in terms of the actual AMC stuff, do you know what the name of the best thing is? Is it the AMC official prep bundle, or like that has everything? Is that what I should be purchasing?
Oh, I mean, whatever would be. So the thing with AMC is that there's a lot that isn't useful because it's from the old
Mm,
hat that they just leave there, so it feels like you're getting a lot.
got it.
Pretty much the question packs, like there's like a bio question pack, physics question pack. That kind of stuff is from the old MCAT. It's not really that useful now, but the cars question packs are still useful because that section hasn't changed. So basically, here I'll show you because I have that open. The AAMC stuff here. Okay, so this is everything. All right.
Yep.
So here, MCAT, section bank, that's what I want you to be doing like this week. So if you hit go to product, you'll see that there's three of them. And you could tell if it's, for the new MCAT by the name, like BB, CP, Psych, Soc. The old
Mm-hmm.
MCAT stuff would just be like physics or bio or like one subject like that. And you see there's like 100 questions each. And, if you hit like practice, it'll show it to you, the different passages and stuff. So that's the section bank.
Mm-hmm.
And then there's okay, the two free tests, and then the practice exams. And of course, you want all the practice exams. But now something like this, biology question pack, don't need that. Don't need that. Don't need that. Same thing, bio question pack volume 2, don't need that. Cars stuff you will need. So cars diagnostic tool, you want.
Mm-hmm.
Chemistry question pack, no, don't want that. Cars question pack volume 1, yep, you do want that. Question pack volume 2, you do want that. Um, and then this question bank doesn't really matter. This official guide thing doesn't really matter. Physics question back here doesn't really matter. And then yeah, section bank, which
Okay.
the same thing as the one up here. So that's weird, but yeah, that's what you want. So basically, you want anything that is involving cars. So the two question packs and the diagnostic tool and the practice exams. And then the only science thing will be the section bank.
Okay. Yeah, that's helpful.
Mhm. Yeah. So that will be, yeah. So I guess you can like kind of do the math to figure out what would be
Mm-hmm.
the best thing to get. I think like each test is like $35 or something, and I think something like some of maybe the question pack for cars is like 15, something like that. But yeah, you don't need a lot of it, but yeah, you want to see what I guess what would be the best price.
Okay.
So yep, so that's for the AMC stuff. But yeah, you're essentially going to be like focusing on that section bank this week, and then we'll go to, full lengths, and then in between will be UWorld stuff. And that can be like, from when you take your full length or when you review it, you'll find areas that you're weak in, and you can do uh questions and stuff on that stuff, on that topic, or those topics.
Okay.
All right,
Got
cool.
it.
So I'm going to share this with you, and I'm going to share also a shared drive with a lot of useful stuff. But let's go over some stuff. Like, would you like to go over like some like material?
Right. So I went over my exam, and so there's a couple
Mhm.
question sets that I would like to touch on.
Yep.
Um, One more thing though before I forget to ask, and when a lot of people they say they recommend to like every time you take a full length, make like a sheet and write like what I did wrong, the correct way to do it, all of that. Is that like worth to do in terms of time, like time wise? Like, because I don't know, to me it's like it feels good to do, but I don't know if I'm exactly learning from it.
Uh-huh. Yeah, I guess you mean like error tracking and stuff, and like,
Right.
I got this question wrong because blah blah blah. Yeah, it depends. I mean, sometimes it might, I mean, if something like that, let's see. Okay. So when you're reviewing your stuff, you kind of already are doing that in terms of making Anki cards
Right.
for the material that you review. So I don't know if it would be the best use of time to start filling in those error sheets and stuff, or error tracking kind of spreadsheets. Um, so yeah, I don't think that would be a good priority if you can, like, if you're reviewing it and you're doing the blind review and stuff and you're making Anki cards, I think that that would be enough. So so that means like you're essentially going to be spending your time, attacking questions and going over material and then remembering the material through Anki. So I don't know if the error tracking is the best tool. I think that it might be a little too time consuming. Um,
Hmm.
it could maybe be useful, let's say, by the end of like, let's say by the end of like your full length, where you're getting like maybe um a small amount of questions wrong. But yeah, I don't know if it would be the most useful. Maybe for cars, but also, we yeah, I would say probably not.
Okay.
Yeah.
Got it. In terms of going over the questions, how do you usually do it? Like, you have students share their screen, you have me send you a screenshot. Like, I just,
Yeah, you can
uh,
share your screen and we can
okay.
go over it. Yeah.
All right, let's do
We
that.
can go over like, yeah, like strategies for how to approach a passage, and that'll include like every opportunity that I get, I'm going to like use to ask you kind of questions about content and stuff, so we can cover a lot in one, like, passage. Uh
Hmm.
that's usually how it goes.
Are you able to see everything?
Uh, yep.
Do you need me to minimize or maximize something, or it looks good?
Um, it looks good. So let's see. Um, okay. So all right. So let's have you do. So yeah, I'm gonna have you read out loud slowly and kind of tell me, or I guess as you read, I'll ask you questions about like so there's something called like buzzwords, which are words that to look out for that they'll likely ask you questions on. So I'll kind of ask you some stuff as we go through this, but yeah, you can start.
Okay. Ovarian cancer is a highly lethal gynecologic malignancy affecting more than 22,000 women annually in the United States alone. Although most patients initially show a complete clinical response to surgery and chemotherapy, recurrent and progressive disease eventually occur in the majority of patients. The sensitivity of tumors to platinum-based chemotherapeutic agents is a major predictor of patients' prognosis. Platinum-resistant tumors are associated with poor outcomes. This underscores the need to identify and eventually target pathways involved in platinum resistance. This task is complicated by the heterogeneity of cells within malignancies. The cancer stem cell theory proposes that resistant cells encompass only a minority of cells within the cancer, but are solely responsible for long-term recurrence. Therefore, chemotherapy must target these CSCs in order to show long-term effectiveness.
Okay, what do you take away from this paragraph?
Um, I mean, the fact that platinum-based chemotherapeutic agents are probably like that's going to be a measure of sorts. The
Mm-hmm.
sensitivity of tumors, like I mean, the sensitivity of tumors to them. Um,
Mm-hmm.
and also, cancer stem cell, basically, yeah, I mean, those are going to be the factors that they use in the experiment.
Yeah, right. So they're talking about chemotherapy and specifically platinum-based chemotherapy, right? And then they say that um, the sensitivity of tumors to the platinum-based chemotherapeutic agents is a good way to predict a patient's prognosis, and that cancer stem cells, CSC, uh, are a small amount of cancerous cells, but are the ones that are responsible for cancer coming back, like long-term recurrence. Therefore, chemotherapy, you would want it to target the CSCs, right?
Hmm.
So, yeah, that's what I would take away from it. And that's, yeah, usually how these types of passages will start out. They'll start out with some type of clinical context, and then from there they'll probably start going into more like the basic sciences that we're expected to know. So, yep, you can continue.
Okay. Aldehyde dehydrogenase activity has been shown to be a promising CSC marker in several types of cancers. Researchers investigated whether ALDH expression in the platinum-resistant ovarian cancer cell line was associated with aggressive tumor behavior. Cells were sorted by whether they expressed high levels of ALDH or low levels of ALDH, and researchers measured invasiveness in response to carboplatin, a platinum-containing therapeutic.
Okay, so this is something you'll see a lot in these passages, which is basically them talking about enzymes, right?
Hmm.
And you can tell it's an enzyme because it would typically end in ASE.
Hmm.
What type of enzyme is a dehydrogenase, though?
Um, it's this has come up in my Anki so much actually, but it adds or removes hydrogens. I'm pretty sure I'm not 100% sure.
So uh so there's different here, let me yeah, so there's
Oh
different
wait, right. So it adds or removes water. It's like condensation and hydrolysis reactions, I'm pretty sure.
so in the chat I'm putting an image
Yeah.
of the different types, the different enzyme classes. And is there a way for you to kind of show that like in your shared screen?
Yeah.
What I sent? Yeah.
One sec.
Yeah. So this is so these are the ends. So like I would make sure to know these enzyme classes and what enzyme classes like things are in. So like one that comes up a lot is a kinase, and that would go here as a type of transferase. But yeah, dehydrogenases are in the class of oxidoreductase, which means that they just have redox happen. So dehydrogenase is a type of oxidoreductase, which is an enzyme that participates in a redox reaction.
Hmm.
So so yeah, that's a good example of one of the buzzwords. Like when you see something like that, or you'll typically see, especially in BB, them talking about some type of enzyme. And a lot of times, they'll do experiments on that type of enzyme. So it's good to be able to, when you see that dehydrogen like aldehyde dehydrogenase or whatever, to know that yes, this is a oxidoreductase and that it's involved in redox reactions.
Hmm.
Yep.
Okay.
Mhm.
Are you able to see it?
Yep. So yeah,
Okay.
aldehyde dehydrogenase, ALDH, that is an oxidoreductase. So it's going to cause some redox reaction to happen. Um, and they're also saying that it's a CSC marker, right?
Hmm.
And that researchers investigated whether the ALDH expression in the platinum-resistant ovarian cancer cell line was associated with aggressive tumor behavior. And that cells were sorted by whether they expressed high levels of ALDH, which they write as ALDH plus, or low levels, which they write ALDH minus for, and that researchers measured invasiveness and response to carboplatin, which is a platinum-containing therapeutic. So, okay, so something that so these BB passages in particular will be very, very much full of acronyms, which can make things really confusing. I mean, we already have CSC, ALDH, A2780, CP70, ALDH plus, ALDH negative. Like that's a lot. These are a lot of acronyms. So what we can do to keep track of them is to write out proportionalities. So um like if something goes up, like okay, so like a lot of people would kind of draw it like this. Like if ALDH goes up, you know, cancer let's say, or CSC or something can go up like that. But I would write that as a proportionality. So I would write like ALDH, and then this is the proportionality symbol. And
Yeah.
that would be proportional to CSC. So, if one goes up, the other goes up. If one goes down, the other goes down. So that's how I would write that. And so, okay, so in figure one here, and for, yeah, the figures and stuff, you want to start with reading the caption. So it says invasiveness and platinum resistance in ALDH positive and ALDH negative cells. And then we have this p-value thing. What's that about?
Statistical significance. And
Mhm.
I guess that's what they, I mean, if you have something that has um those two, like, whatever you call them, asterisks, right?
Mhm.
Uh, came out of my head. But yeah, that means that something is meaningful, that there is a statistical significance.
Yeah, yeah. If it says a p-value, like if it's less than 0.05, that's statistically significant. And if it's less than 0.01, it's of course less than 0.05, so that's statistically significant. But only because of the p-value, because they could give you asterisks that will highlight different things.
Hmm.
So it's just because of the p-value that we know that it's statistically significant. But good, good. So okay, so now let's take a look at the figure. So there's two of these, right? So let's look at A. What is A showing us?
Right. So I think one issue, if I remember correctly, during the exam,
Hmm.
it was like I had a decently hard time understanding what exactly meant to be invaded. Um, so
Hmm.
cells invaded. Now, I mean, I don't know, is that maybe how, like, basically it's seeing how respondent they are um to the platinum. But actually no, that's the second ref. Hold on. So it's a cancer cell line, and then the enzyme ALDH is being acted. And so when there is a ALDH, there is a statistically significant amount of um, cells that were. I didn't know what it meant by cells invaded.
Hmm. So
Like,
let's think about it like this then. Do you think it's good or bad?
I think it's good because it's invading cancer cells.
So let's think about how ALDH itself. If you have a lot of ALDH, um,
Oh wait, no. If yes, okay, it's a CSC marker.
mhm,
So there's more. If it's invading more cancer cells, there's more cancer cells.
mhm, yep. Yeah. So yeah, if it's not clear right away, I would go into like, is this something that's likely to be good or bad? And I think that they're trying to tell us that, yeah, exactly what you said, that it's a marker for CSCs, and CSCs are not good. So cells invaded probably means not good. Um, and also, if I think about the word, it sounds like it's bad, invaded. Like
Yeah.
that's not like, enough for me to, definitively say, but it sounds like it's not so good. Um, so yeah, I would, you know, uh write so this proportionality symbol, and maybe I'll do this. I'll write ALDH plus and then invasiveness. it's a little, you have to be a little careful here because it looks like, I mean, typically ALDH plus or something plus and something minus would mean plus has it, minus doesn't. But is that the case here?
No, minus means low levels.
Yeah. So we want to make sure that we understand that. So yeah, ALDH plus just means high levels, and then ALDH minus means low levels. Good.
Um,
Yep.
Before I forget, what is your highlighting strategy though? I mean, not maybe not strategy, but like
Uh-huh.
I should be highlighting a decent amount, right, when I'm doing every passage
Uh
in BB.
so I think that I would write certain things out like these proportionalities here um instead of highlighting. I mean, you could certainly highlight if it helps with reading. Like sometimes it can help someone like kind of read a passage by highlighting as they go through it. Uh,
Yeah.
I don't have like any real like set highlighting strategy. I have more so like I would write something like this down, like ALDH plus is proportional to invasiveness because it's really hard to, again, like you're gonna see so many acronyms. It's really hard to keep track of all of that, even if you have it highlighted. It's really just hard to keep track. So the proportionality stuff you'll see me do a lot more of. But yeah, I don't have any real set kind of highlighting strategy. In cars, I do, but that's more so in the way that I teach cars, less so that something that I would specifically say to do on the exam. But yeah, if you feel like it helps you read by highlighting stuff, then you can definitely do that.
Thank you. Okay, that makes sense. Um, okay.
Yeah. So okay.
Now in regards, yeah, second graph.
Mhm.
Um, so yeah, so I remember I was running out of time because I saved this for last this passage, but um, I was just having a little difficulty understanding what sham was.
Okay. So
Yeah, so
uh-huh. Got it. So
was yeah, I'm listening. I'm listening.
sorry, yeah. So what do you what's B supposed like what are they saying B like figure 1B is representing?
So they introduced carboplatin
Mm-hmm.
into into the experiment.
Uh-huh.
Um, However, the dependent variable changes.
So so we do say response to carboplatin here, but here we say platinum resistance, so that is also a way of seeing what 1B is, right?
Uh-huh.
So okay, so it seems like we have so what's different here is that we have two for each of these kind of conditions. We have ALDH minus and ALDH plus. Um, so we can make like a direct comparison between, like with having low levels and high levels. Um, but if this is supposed to measure platinum resistance and the carboplatin is the platinum containing therapeutic, as I say here, then sham would have to be something that doesn't contain platinum, right?
So just a control. Okay.
Yeah. And then, have you heard of
Okay.
that word before? Like when someone says like something's like a hoax or made up.
Yeah, yeah, that's the thing.
Yeah.
I had a feeling it was that, but
Yeah.
I just haven't, I don't hear that word in like
Yeah.
common
Yeah.
daily language.
Yeah, that's another thing. Like a lot of times when I ask students like what a word means, they think that I am asking them like what the like a dictionary definition is, but most of the time it's going to be a word that you've heard before. And if it's in a different context, it's fine because it probably like is going to be analogous in a different context. So yeah, if you see a word that you've seen before in a different context, it could still be valuable. So
Hmm.
so yeah, it's exactly that. So yeah, so we essentially see that in the sham treatment, which must be some thing that they put in that doesn't contain platinum. Uh, we have a lot of, well, both, ALDH minus and ALDH plus is a lot more or higher in terms of, I guess, colonies per field, compared to when the carboplatin is involved. So what can that tell us about, I guess, yeah. So yeah, what can we take away from that?
That the carboplatin, that platinum containing therapeutic is detrimental towards the tumor.
Uh-huh. So yeah, we see that. Okay, so like this goes like down like that, right? The darker bar goes down, and the lighter bar goes down too. But when they're giving us the asterisks here, what are they kind of comparing? Like, are they, because like here, this would be comparing it to that, right? So what would this be comparing it to?
Um, yeah, I don't know because I'm not sure if it's between um I don't know if they're comparing the ALDH pluses or if they're
Yeah. All right, how about this then?
comparing ADOS
So
minus and ADOS plus in each one.
yeah. So okay,
I
so
would think it's the gray lines because there's only one variable involved, so to speak.
yeah, so all right, so it seems like okay, so if I look at the dark bar, that goes down when you have carboplatin. When I look at the light gray bar, that goes down as well. So it seems like that would mean that carboplatin is doing its job in killing these cancer cells, right?
Hmm.
But I think that what the asterisks are comparing is, I guess, how much it goes down compared to for ALDH plus compared to minus. And we can see that although both bars went down when you have carboplatin, the ALDH plus bar does not go down as much as the ALDH minus bar,
Hmm.
right? So I think that would make it so that ALDH plus would be proportional to platinum resistance, because although it goes down, it does not go down as much as when there's low levels of ALDH, right?
When you're saying it goes down a lot, you're comparing the two gray bars.
Oh yeah, so
So the ALDH plus, both of those.
I'm comparing it to the darker bar because
Oh, okay.
if I was comparing it to just the gray bars, it would seem like, this gray bar is here and this one's here, and that seems like it goes down, right? So if I did,
Hmm.
so like let's pretend actually then, like let's pretend that I didn't have, um, let's pretend I didn't have the dark gray bars like this, right?
Hmm.
This was the case. I the only conclusion I could reach is that carboplatin works because in the sham I have a high number of colonies per field. Oh, and by the way, like colonies per field, like what does that mean, right? What do you think that means?
How much, how many cells are growing?
Yeah,
Like the tumor amount,
yeah. So yeah, something bad, right?
right?
So so if I didn't have those dark bars like I have right here, because I whited them out, it would seem like carboplatin is doing what it's supposed to do. It's killing cancer cells, right?
Okay. Yeah.
But since I have these dark gray bars, now I can see that compared to the dark gray bars, the light gray bars are higher. So now I can't I can't really say that carboplatin is doing its job. I mean it is, but since I can now compare it to the ALDH minus, it's not going down as low as it does for ALDH minus. So this shows me that ALDH plus has some has some platinum resistance to it. Because if it didn't, it should go down as much as the dark bar does,
Okay.
right?
That makes it a little more clear.
Yeah. Because if you just had the gray bars, it would be like, okay, it's working. It's not platinum resistant, right? But now we can say that it is platinum resistant because we get to compare it to the ALDH minus bars.
Hmm.
Yeah. These BB like kind of figures and graphs and stuff could be really like a nightmare. So I remember it took me a long time to be able to make sense of them. But that's what I would do though. Like I would like try to make sense of them and I would write what I wrote here with proportionalities. So I can understand the relationship between high levels of ALDH and invasiveness and then high levels of ALDH and platinum resistance. Yeah. How do you feel?
Look good. I mean, makes sense.
Okay.
A little more now.
Mhm.
Yeah.
All right.
Okay. So in a subsequent
Once-
experiment, oh, no problem.
All right.
Okay. Um, in a subsequent experiment, targeted short hairpin RNA, shRNA, was used to silence ALDH expression. Invasiveness and percent survival in response to carboplatin treatment were measured in control, SH control and ALDH knockdown cells. Um,
Yeah.
okay.
So see, there's more and more acronyms, and that is a lot. But let's see, right? Okay, um so they're using this type of shRNA to silence the expression of ALDH, and then they measured invasiveness and percent survival in response to the carboplatin treatment in the control group and then the knockdown group, right?
Hmm.
And and then the figure says invasiveness and platinum resistance in the knockdown cells, and then same thing with the p-values. And then yeah, what does figure 2a say or show us?
Okay. Whew. Um, well, first of all, there's no statistical significance because the two the two things are like they're overlapping.
Yeah,
Um,
perfect. Yeah, that's really good to know. Like even if they didn't give you the asterisk stuff, if you have error bars that overlap, yeah, there's no statistical significance. Good job. So yeah, so it seems like ALDH doesn't seem to be playing too much of a role here in invasiveness. All right.
I have a question though. Doesn't that kind of go against what
1A.
1A was saying, or 1A was just a different experiment over just in general?
Good. Yeah. So yeah. Good that you noticed that, because yeah, it would contradict 1A. So my thought is that maybe it isn't merely the presence of ALDH that causes cells to be invaded, but a high amount compared to a low amount.
Mm,
But I like how you caught that.
right.
Good.
Okay, next, right? The last paragraph.
Oh,
The exp
QB.
Oh, okay, yeah, yeah.
If it's hard to see, you could zoom in if you want.
Yeah. But it's showing that carboplan has a statistical significance on the amount of survival. Like in when there's ALDH that's like removed. Um yeah.
Sorry, yeah. What were we saying about it?
So that carboplatin um has a significant effect on the survival of the cells
Mm-hmm.
in the cells that have ALDH that's silenced.
Mm-hmm.
Um
So when they silenced ALDH, um, less of the cells survived, right?
mhm.
Um, so that's kind of consistent, I guess, than what from what we saw earlier, right? Where we saw that high amounts of ALDH caused, you know, less less, or it caused more platinum resistance. So here we see percent survival control is higher than when you knock down ALDH. So ALDH would be playing a role in survival, right? And then if you increase the amount of carboplatin, both bars go down, but you still have the silenced ALDH one that's lower in percent survival, right?
Mhm.
So, and now it kind of helps us a little bit with the context, because in 1B we had, like, we're comparing low levels to high levels, and in 2B we're actually now comparing like the presence of it and then the absence of it. So it's not necessarily the amount here, but they do make the amount in terms of carboplatin. But yeah, so essentially we see that when you have a higher amount of carboplatin, that survival goes down for both control and silenced, but it's going down more for the silenced one. So that would mean that ALDH, like the presence of it, I would say would be proportional to survival. Survival, or rather, like, it's survival, but so here it's colonies per field in the y-axis, but it was there to measure platinum resistance, right? And same thing with this, it says percent survival, but it's there to measure platinum resistance. So I would really then just say that the presence of ALDH would be proportional to platinum resistance again. And I did separate like ALDH here from ALDH plus, but it's essentially having similar results. So yeah, so it's kind of like this in that both of these go down, but comparing the control that has ALDH and the silence that doesn't, the one that doesn't goes down more. So that would mean that ALDH helps the survival, which would mean, in other words, more platinum resistance, right?
Mhm.
Okay.
In a way, this is kind of like it's a little bit like it's hard to fully map everything out in my brain if that makes sense.
Yeah. It'll help as you like continue to do this proportionality stuff because because I haven't actually even shown you like an inverse proportion yet, but yeah. If it helps to write maybe in parentheses like colonies per field or survival, percent survival, then that's definitely something you could do. But yeah, pretty much that's what we're seeing so far.
Hmm.
Yep, you can continue.
The expression levels of genes in the ALDH knockdown cells were analyzed using polymerase chain reaction. A significant increase was found in the expression of BAX, a pro-apoptotic gene, and a significant decrease in CDK4, which encodes a protein that promotes progression from G1 to S.
All right. So again, more acronyms, right? But now this can help us in terms of setting up like an inverse proportion. So, okay, so they found so okay, they looked at, oh, by the way, what's meant by expression?
How much, I mean, how much, like, how much the genes are being translated
Yep.
in the cells that have no ALDH.
Yeah, good. Yeah, you'll see expression being mentioned a lot, and that's like because like DNA can have a gene for something, but in order for it to be expressed, it has to like be transcribed into RNA and then translated into the protein, right? So good. Yeah, so they looked at expression of genes in the knockdown, ALDH knockdown cells, and they found what do they find?
There was an increase in the expression of a gene
Yep.
that leads to apoptosis.
Yeah, right. So they found so in the knockdown ALDH, they found an increase in BAX, right? Which is pro-apoptotic. Now, I would then put that as an inverse proportion, which is done by putting it under 1. So 1 over BAX. And that's why, like, because again, I guess I didn't mention this before, but a lot of times people like to write something like this goes up, let's say this goes down, or something with arrows like this, but that's not what you want to do. You want to write it proportionally like this because they'll likely ask, like a lot of times they like to ask what a graph of it would look like.
Hmm.
And if both things were going up, like let's say A is proportional to B, that would just be like this, right? This like linear kind of.
Hmm.
But what would an inverse proportion look like?
Like, I mean, it would be like a exponential sort of Like
Okay. How do you know that? Like, why wouldn't it just be?
because it's approaching
Yeah.
a certain value,
Yep.
which is I don't even know what,
Because what
but
number?
zero.
Yeah. Right. Because B can't be it. You can't have 1/0 would be undefined, right? So
Yeah.
the axes are asymptotes here, and yeah, it would look like that. Good job.
Hmm.
So yeah, so we say that, so they're saying that when you don't have the ALDH, that you have an increase in backs. So I put like ALDH proportional to 1/backs to indicate that inverse proportion. Because if you don't have ALDH, you have backs. So I could have alternatively written it like this. Oops. One like over ALDH is proportional to backs.
Yeah.
But since I already had ALDH, I'm just gonna put inversely proportional to backs. Which would be proportional then to, or would it be proportional or inversely proportional to apoptosis?
It would be proportional. You're saying
Oh yeah.
ALDH, it would be inversely proportional to APOP just because backs codes.
Yep. And you'll see that a lot in when passages talk about cancer. Like cancer will have like, like the two-hit hypothesis or whatever thing, like uh an increase in the production of like tumor cells and a decrease in apoptosis that would lead to cancer. So we see that when you don't have ALDH, that there's an increase in backs, which causes apoptosis, which is what a cancer cell isn't going to likely do. So it makes sense that if you get rid of ALDH, which so far we've been saying, is something that's present in cancer stem cells, that yeah, if you get rid of that gene, then you have a higher amount of backs, which means a higher amount of apoptosis, right?
Hmm.
Yep. Okay. They also found a decrease in CDK4, which encodes a protein that promotes progression from G1 into S. CDK4, would you say that that's proportional or inversely proportional to ALDH?
Proportional.
Good. Kind of ran out of room here, but I'll put it like, I'll just put it here. So
Hmm.
CDK4. Cool. Okay. So good. All right. Is there anything underneath?
No.
Okay. So yeah, there we go. So that's essentially what I would take away from this. I would take away that ALDH is present in cancer stem cells, and high amounts of ALDH would give you higher amounts of invasiveness and higher amounts of platinum resistance. And the mere presence of ALDH still would indicate platinum resistance. And if you don't have ALDH, then you're going to have more backs. And that would mean, more apoptosis. But if you do have ALDH, it would mean more CDK4, which would mean more cells go from G1 to S, which basically means like more cell division, right? So yeah, that's pretty much all I would write for this passage.
Can we go over graph 2b because
Mhm.
I don't have a fully clear understanding of it,
Mhm.
and I would like to
Yep.
look at it again.
Yeah. So yeah, tell me what your, I guess, what you're thinking on.
I'll just I'll let like my words flow freely. But so basically, percent survival, the higher it is, the more resistant they are to carboplatin.
Mm-hmm.
Um, Now we see that as you increase carboplatin, they have like less are surviving.
Mm-hmm.
Um, s- so I'm trying to understand like because there's like multiple variables at play here, and it's just like I feel like in a time situation. I know as you're saying, you have to do the relationships. It's just like a lot. It's like sensory overload, so to speak. So
Yeah.
I'm also trying to understand the relationship in terms of like the statistical significance, how that plays into it.
Mm-hmm.
Um,
Yeah. So let's say we had, all right, let's say we had, okay, let's say just we just had the black bars, then it seems like as we increase the amount of carboplatin, there's less survival, right? So more of these cancer cells are dying.
yeah.
And if we do the same thing with the black bars, we could say if there's more carboplatin, there's less survival too, right?
Hmm.
Now, if we leave both bars, we can then make a comparison between the control that has ALDH expressed and the silenced one, which doesn't have ALDH expressed. And then we can make a comparison, unlike before, which showed us that carboplatin is working, it's doing its job, it's reducing survival. But now we get to see what effect ALDH has on this amount that survives with increasing amounts of carboplatin. So we could see that in the first thing over here, we can see that with the same amount of carboplatin, that there's more survival in the control, and the control has ALDH. So
Yeah.
ALDH means more survival. If you compare these two. And if we increase the amount of carboplatin here, both bars go down, but one goes down more than the other. So here we see that the silenced ALDH, the version that doesn't have ALDH expressed, there's less survival for it. So, oh, this would be a good idea. So let me do this. Let me do survival is proportional to platinum resistance. And we would see that um, that SH control in terms of percent survival it would be larger than SH ALDH1, which means that um which means that the amount so essentially if we have more cells surviving in the control condition compared to the one that doesn't have ALDH, then that can help us understand that ALDH is playing a role in the platinum resistance since this one has it and this one doesn't have it. So that's what we're trying to compare here. Because if we didn't have, like, if we were not comparing like these two graphs, then we would say that that if you have carboplatin, that there's less survival. Same thing with 1b here. We see that with carboplatin, this one goes down and this one goes down as well. Same thing here. With more carboplatin, the black bar goes down and the gray bar goes down. So, that just shows us that platinum is causing less survival, causing less whatever colonies per field, which we're not denying. But if we want to make a comparison between, not between sham and carboplatin, but between this black bar and this gray bar, that's what we're trying to do. So for figure 2b, like we see that when you have ALD, so like let me call it SH control ALDH1 and silenced one, just like no ALDH, I guess.
Yeah.
We know that if it has ALDH, it's going to be more likely to survive when you give it a platinum containing therapeutic. And if it's more likely to survive, it's going to be more resistant to the platinum.
Yep.
So
That bridged it a little. Yeah, that's good.
yeah. It's tough because there's a lot going on, but yeah, if you just had, like, I would think about it like this. Like if we just, because this could be a good way to go about it, like, because we have, I guess, a lot of things that we're comparing. So I would first think about it like in terms of the black bar. And I see that black bar with 50 micromolar carboplatin is higher than 100 carboplatin. So carboplatin is killing these cells, producing less survival. So let's say platinum is inversely proportional to survival, right?
Yeah.
And I see that if I look at the black bar, the increase in platinum, less survival. In the gray bar, increase in platinum, less survival. So platinum inversely proportional to survival. But when I compare the black and gray bars, I see that with the same amount of platinum, that the control has more survival. And the control is the one that has ALDH. So I could say that if something has ALDH, it's going to be more likely to survive. And we can then link survival to platinum resistance. And so we see that although the increase in carboplatin caused less survival in both control and silenced ALDH, that the control is still going to be having a higher level of survival than the one that doesn't have ALDH.
Okay.
So essentially what we're seeing here is ALDH contributes to invasiveness as well as platinum resistance.
Okay.
Because although the platinum causes less survival in both cases, it's not as low as it would be if it didn't have the ALDH.
All right.
Yeah.
Should I go to? I got this question right on the exam. I mean,
Okay.
yeah, I'd rather go over this before anything.
Mhm.
Um,
All right. Yeah.
which of the following? Yeah, I
Yeah.
should go. Okay. Which of the following conclusions is most strongly supported by the data presented in the passage? ALDH expression is associated with additional factors that promote invasiveness distinct from platinum resistance. Platinum resistance is the mechanism through which ALDH promotes invasiveness. ALDH expression is exclusively responsible for the platinum resistant phenotype. Back's expression is promoted by ALDH.
All right. So how would you typically go about this?
I mean, after reading the answers, I guess we could pretty quickly eliminate D. Because I mean, when you do write it out, the relationships, it's obviously makes things a lot easier.
All right.
So yeah, after that, um, it, it wouldn't be B.
All right, tell me why.
I think those are both they're both dependent variables
Yeah.
that we like, yeah, because in 1A and 1B, one of them was measuring invasiveness and one of them was measuring platinum resistance.
Mm-hmm.
So like B is saying that the two different things we measured are like um yeah, I don't even know. I'm like getting jumbled up in my own words, honestly.
Yeah, I think that so it's saying that um the mechanism through which ALDH promotes invasiveness is platinum resistance. And that could be the case if we look at figure 1, because figure 1 shows that ALDH causes more invasiveness. That's figure 1A. And then figure 1B shows more platinum resistance, right?
Mhm.
Now, if that was the only data that we had, B would be possible. But we also have figure 2. And figure 2 shows that there isn't an increase in invasiveness. So, but we still have an increase in platinum resistance for 2B, right?
Okay.
Like if we just focus on figure 1, we see, oh, okay, ALDH, more ALDH means more invasiveness, and more, ALDH means more platinum resistance. But figure 2 shows that there isn't a change in the invasiveness. So, but there is, a difference in platinum resistance. So we can't say that platinum resistance is the mechanism through which ALDH promotes invasiveness because we have platinum resistance happening without a difference in invasiveness happen.
Right.
So B is not true because of 2A specifically.
Okay.
Oh, yep. So we can get rid of that.
I don't know. See, the thing is,
Look, I'm gonna be completely transparent with you. From what I remember, A was the answer.
Okay,
So like, I'm also not like I still have that bias.
I see.
Like I can't, like I can't fully just I am trying to understand right now though why C is wrong.
Oh yeah.
Um, because I just remember like the A was the answer.
Mhm.
Um, when they say it's exclusively responsible, like didn't it say that the targeted short hairpin RNA was used to silence it? Does that have anything to do with why C is wrong?
Uh-huh. Yeah. So okay. ALDH expression is exclusively responsible for the platinum resistant phenotype. So that's saying that there's only one thing that causes platinum resistance, and it's ALDH expression. Now, if we can find an example of anything else possibly causing platinum resistance, C would have to be wrong because they're getting kind of extreme with this word exclusively.
Yeah.
And that's like whenever you see like if you see like extreme word, which means if you were to assign like a like a percentage to it, if it's like 0% or 100% or like 1% or something, if it's like only, because
Mm-hmm.
is saying the same thing as like only. So
Yeah.
yeah, so those you want to just always be careful when you see words like that. But but yeah, if we can find a reason or yeah, if we can find platinum resistance in something that doesn't have ALDH, then we would be able to prove C wrong. So I would look at ALDH, control compared, sorry, ALDH, knockdown. That's what I would look at, and I see that percent survival for control is 75% when you have the ALDH, and then when you don't, it goes down to 50%. Um, so I guess something I would be looking at or thinking about is, uh, I guess, why wouldn't this control be, I don't want to say like, why wouldn't it be 100% or why would, no, you know what? This would be better. Not why this isn't 100%, but why is this not like 0%? Right? If this was the case that ALDH expression is the only thing that's causing platinum resistance, when we get rid of it, why do we still have some survival, which would mean some platinum resistance, right? If it was the only thing that caused platinum resistance and we got rid of it, we should see no platinum resistance, right?
Mm-hmm.
So we can't say that ALDH expression is the only thing that causes platinum resistance, because if that was the case, when we get rid of ALDH, why do we still have resistance, right?
Yeah.
So that's how I would uh evaluate C. So
Mm-hmm.
a lot of times it has to just do with kind of wording it in a way that makes sense to us.
Mm-hmm. All right.
Yeah. And then so I guess A says, and you see how A is a lot more vague. It's saying
Yeah.
associated with additional factors. In general, the more vague you are about something, the more difficult it is to prove wrong, right? Because like C, exclusively responsible, is easy to prove wrong. You just need like one counterexample, but associated with additional factors is so vague that it's harder to prove wrong. Because it's saying, and C and A are also saying kind of like the opposite. C is saying platinum resistance is only caused by ALDH expression. Whereas A is saying platinum resistance is caused by ALDH expression as well as some other stuff. And that some other stuff could be anything and therefore is more difficult to disprove. But I want you to see it that way. I want you to see that C is saying platinum resistance is only through ALDH expression, and A is saying platinum resistance is through ALDH expression plus some other stuff. So maybe if I wrote ALDH expression equals platinum resistance versus ALDH expression plus some other stuff equals platinum resistance. So either ALDH expression equals platinum resistance or ALDH expression plus stuff.
Yeah. Okay.
Yeah.
And then this was the other question.
Okay.
If true, which of the following findings would that most directly challenge the result of the study? ALDH expression correlates with the expression of other determinants of tumor invasiveness. Targeting a different pathway instead of ALDH yields better improvements in platinum sensitivity. No significant relationship is found between ALDH expression levels and clinical behavior in non-carboplatin-resistant ovarian cancer ovarian cancer cell lines. ALDH overexpression results in no significant differences in platinum resistance compared to control cells. Okay.
So what are we looking for here?
I mean, I should probably connect ALDH and platinum sensitivity.
Okay.
I guess we can eliminate A.
Let's see. ALDH expression correlates with the expression of other determinants of tumor invasiveness. So we're trying to find like what would weaken the results of the study, right?
Mm-hmm.
Okay. Oh, and like what would directly, I guess, weaken? So would it weaken it if we find that ALDH expression correlates with expression of other things that determine whether a tumor is invasive or not? Would that go against, like, would that directly disprove anything here?
Not really.
Tell me why.
I mean, I think the passage itself was talking more about not only invasiveness but like platinum resist sensitivity. Um,
So this is kind of okay, so this is kind of like the other kind of choice that we had where we talked about the invasiveness and relationship with that and ALDH. Because if we just looked at figure 1A, we would see that more ALDH would mean more invaded cells, right?
mhm.
But figure 2A where you don't have, you either have the ALDH in the control or you don't in the silenced, have the same amount of cells invaded, right?
Yeah.
So figure 2A shows us that invasiveness must be due to something other than ALDH. Right? Because otherwise, we should see a difference in the amount of cells invaded when you have the control, which has ALDH, and the silenced one that doesn't, right?
Yeah.
So okay. So A says that ALDH expression could be correlated with the expression of other things that determine invasiveness. That wouldn't directly challenge the results because that's what we see here, right? Because if this is showing us that ALDH is not solely responsible for the invasiveness, so it must have something to do with some other things that could also happen, right?
Yep.
So A, it would be kind of supported by figure 2A, that there could be some other determinants of invasiveness. So yeah, that wouldn't directly challenge it. So yep, so we can get rid of that. What about B?
I mean, again, I don't know if like the passage isn't really saying that AODH is like the best at what it does.
Yeah.
So I don't see how B would like most directly challenge it.
Mm-hmm. Okay.
So yeah, I don't know. That's my thought process.
Mm-hmm. So yeah, so B is saying that it would like if it's the case that there's a different pathway instead of ALDH, that would give you better results in terms of platinum sensitivity. Um, you know, I don't think it would refute what the passage is saying, which is that ALDH plays a significant role in platinum resistance. If something else gives you similar results or better results. So yeah, so maybe that one won't be it. And then let's look at C.
Is that like is that saying the same thing that's shown in 2A? Oh wait, no, it's not. It contains talks about carboplatin, non-carboplatin resistant. Mm, is it saying what's in 2A?
Let me see. No significant relationship is found between ALDH expression levels and clinical behavior in non-carboplatin resistant ovarian cancer cell lines. Um, so I think that they were only looking at this. Okay, so they're using this platinum resistant cancer cell line And so C is talking about if they used non-resistant cell lines, cancer cell lines. So it's different from what we have. But if it was true that there's no relationship between ALDH expression levels and, I guess, something with non-carboplatin resistance cancer, would that, I guess, directly challenge the results of the study?
No.
So something that could also help is for like weakeners, take like, I mean, this usually helps the most in cars, but could still work here. Like if you take the main idea and you flip it, what would agree with that, right? So I think the main idea for this passage is ALDH expression or high amounts of ALDH expression result in platinum resistance. So what would most directly challenge that? Something that would say that high amounts of ALDH expression does not cause platinum resistance. So that's a way to go about weakener questions, which this question is because it says challenges the results of the study.
Okay.
So I think so D says ALDH overexpression results in no significant differences in platinum resistance compared to control cells. I think that is what is directly challenging the results of this study. Is that the answer?
I actually don't remember this one, but I mean that's what we were talking about like for the past hour almost, right? Like the idea that when you do have a lot of ALDH, it leads, yeah, I mean it leads to platinum resistance.
Yeah. So this is saying higher amounts of ALDH means more resistance, platinum resistance, and this is saying like the mere presence of ALDH means more platinum resistance. So overall, the passage is the results of the study would be that ALDH increases platinum resistance and amount of ALDH increases platinum resistance. So yeah, what would directly challenge that is if we found that if we have more ALDH that we don't have a significant difference in platinum resistance.
Okay.
So yeah, I would take like the main idea and flip it and then look for that.
Okay. Sounds good. I do see we are running out of
Oh
time.
yeah. Yeah.
So
So
it
yep.
is your next session in 2 minutes?
No.
Because I was going to ask like before. I mean, first of all, two questions. First of all, in terms of payment, I mean, do you accept Zelle?